edition is okay. And this is the enhanced version of the same book, 11th edition also, called Burton's Microbiology. The learning objectives of this lecture and next lecture to go into certain definitions, phototrophs, chemotrophs, autotrophs, etc. We'll go through them. We talk about enzymes, catabolism and anabolism, biochemical reactions, including aerobic respiration, glycolysis, and Krebs cycle, and electron transport chain, oxidation reduction. And then through genetics, we are going to go through what's the meaning of beneficial, harmful, and silent mutation, and to go describe the different ways of bacterial genetics, lysogenic conversion, transduction, transformation, and conjugation. These are the themes of the two lectures. Starting with microbial physiology. What is microbial physiology? Is the study of the vital life processes of microorganisms. So we are talking here about physiology of microorganisms. As you know, we talk about human physiology, animal physiology. Also, microbes have also a physiology. Microbial and nutritional requirements. Living protoplasm contain 6 major chemicals. It means that this microorganism or the microbes in general require 6 major chemicals. The main one is the carbon, hydrogen, oxygen, nitrogen, phosphate, sulfur. Other also sodium, potassium, chloride, magnesium, calcium, iron, iodine. Uh, with these chemicals, the microbes are able to make up macromolecules. تصنع macromolecules اللي تحتاجها. However, essential amino acid and essential fatty acid are not synthesized by microbe and should be supplied to the microbes in order to grow. Should get these materials from the environment. Now, how we categorize the microorganisms according to their energy and carbon sources. As you know, microbes should get energy and should get carbon. We said carbon is very important material for the growth of microbes. According to the energy and carbon, we can subdivide the microbes in general. Let us talk in general into 4 categories. The word photo from the beginning, it means light. Chemo, it means chemical. Auto, it means requirement of, they get the carbon requirement from CO2. Hetero, it means they get the carbon requirement from organic materials. So let us go into photoautotrophs. Photo, it means light, CO2, trophs. Photoheterotrophs, light, chemical, that is organic material, photoheterotrophs. Chemo, from chemicals, organic, CO2, and the trophs. Chemoheterotrophs, chemical, organic material, a troph is the living microbe. So 4 categories. We will go into details of these microbes in a moment. What's the meaning of phototroph? It means light as energy source, photo. Example, photosynthesis. التصنيع، التمثيل عن طريق استخدام اللايت means conversion of light to energy. Energy, we will see how energy is mediated by ATP, of course. Chemotrophs, inorganic or organic chemical as source of energy. Chemo, organic, inorganic. Inorganic are called chemolithotrophs. For simplicity, we say lithotroph. Microbes that use inorganic materials are called lithotrophs or chemolithotrophs. If they use organic material, organotrophs or chemoorganotrophs. We are going through definition, as I said. We are going to depend on these words later on. So you should know what's the meaning of chemotrophs, phototrophs, and so on. Now, regarding the carbon source, from where the microbe gets the carbon, if we say autotroph, it means they use CO2 as the source for carbon. Example, plants. تعرفوا البلانت تاخذ ثاني أكسيد الكربون cyanobacteria. If I tell you from where cyanobacteria get the carbon, you will say to me from CO2. Why? Because they are autotrophs. Hetero, we said they use organic compounds, not CO2. Example, human cells, fungi, protozoa, most bacteria. Remember this word here. Most bacteria are heterotrophs. This should be clear. There is another word, you should know the definition, ecology. What's the ecology? Is the study of interaction between organism and the environment. For example, autotroph, photo it means light, auto CO2. Autotroph produce food and oxygen for what? For chemoheterotrophs, as if they are the manufacturers for the chemoheterotrophs, المصنعين الغذائيين للchemoheterotrophs. Now let us combine things with each other regarding energy and carbon source. Photo, it means light. Auto, CO2. Example, plants, algae, you know algae. You have studied it in the beginning of the course. Algae and cyanobacteria. Photo, heterotroph. Photo, light. Hetero, organic materials. Some bacteria are photoheterotrophs. Chemoautotrophs, chemo, chemicals. Auto, CO2, certain bacteria. Now we have chemoheterotroph, chemochemicals, heteroorganic material. All animal cells, human cells, are chemoheterotroph. Fungi, protozoa, and most bacteria. If I ask you a question, most bacteria are what of these? Are chemoheterotrophs. You should know that most bacteria are chemoheterotrophs. That is to say, they use chemicals and organic materials. Chemicals for energy, and organic material for CO2, for carbon source. Okay? This table summarizes to you what I have said. Just I put it for making things simple. This is what I have described already in the slides. Now let us talk about metabolic enzymes. Metabolism, chemical reaction, and the reaction are called metabolic reaction. Metabolic enzyme control these reactions, and these enzymes under control of genes. Remember that enzymes of microbes are under control of gene. دائما الانزيمات تحت سيطرة الجينات. Enzymes are biological catalysts. What is the meaning of catalyst? المحفزات. تحفز الreaction. The enzyme are quite specific. They act on one substrate. Each enzyme act on one substrate. It is like lock and key, زي القفل والمفتاح. شو معناته؟ يعني هذا القفل، هذا المفتاح يرهم على هذا القفل. Lock and key. And the enzymes, they do reaction and then separated and go unchanged to share in another reaction. يعني الإنزيم يشترك بهذا الرياكشن وينسحب unchanged، لا يتأثر، لا يتغير، ليشارك مرة ثانية وثالثة ورابعة في reaction أخرى. The same reaction. The enzymes are either endoenzyme within the cell or exoenzymes produced within the cell then released outside the cell. يعني تصنع داخل الخلية وتفرز خارج الخلية. released يعني تفرز خارج الخلية. cellulase example, pectinase of saprophytic fungi. I am giving you basic things, ABC of microbiology. So we are going to use these terms in the future when go through the course. saprophytic bacteria, saprophytic fungi, الفطريات الموجودة في الانvironment. Look, this is the substrate, this is the specific enzyme. It is like lock and key. So they bound very specifically. The enzyme now will act on the substrate, separate it into S1A and S1B. تقسم هذه المادة إلى قسمين S1A, S1B. And then the enzyme will be separated and go to share in another reaction, unchanged. We are saying something basic. The factors that affect the efficiency of enzymes are affected with many factors: pH of the environment, temperature, concentration of the enzyme and the substrate, the presence of metals. Some of metals increase the activity of enzymes, like what? Calcium and magnesium. So calcium and magnesium enhance the action of enzymes. Some metals inhibit the effect or efficiency of enzyme, like what? Like lead, zinc, mercury. And you know these metals. So metals, some of them enhance the activity of enzymes, some of them inhibit the activity of enzymes. So as I said, sometimes we can cheat, نخدع the enzyme. We put a substrate similar to the original substrate of the enzyme. That is to say, mimic, يعني يشابه, mimic, يشابه the original one. So we can block the reaction wanted by the microbe. يعني ممكن نحط أو نضع دواء يشبه السبستريت، فالإنزيم سوف يخدع فيعمل على هذا الدواء بدل الأوريجينال سبستريت، فالناتج راح يكون false. So the microorganism will be killed. This is the basis of some chemotherapeutic agents. بعض الأدوية، بعض المضادات التي تعمل بشبه السبستريت فتخدع الانزيمز وتعمل عليها بينما هي مادة مشابهة للأوريجينال سبستريت، فالناتج راح يكون غير صحيح، وبذلك المايكرو أورجانيزم سوف تقتل أو تحبط. Chemotherapeutic agents. So we are making use of this characteristic on how the enzymes work on the substrate. We are going to come to this point later on. When we will talk about antibiotics, inshallah, in the coming lectures. Metabolism, as I said, could be catabolism and anabolism. Catabolism, a breakdown of large molecules, break the bonds. When you break bonds, energy will be released. On the opposite, anabolism, anabolic reaction, reassembly of a smaller molecule, creation of bonds. This require energy. So they take energy and stored in the bonds. So catabolism is opposite the anabolism. And you are going to see this in table, and you should know that catabolism and anabolism are in equal or in balance. قديش عندك catabolism قديش عندك anabolism. So ATP can be transferred the energy. Catabolism release energy. Anabolism require energy, consume, تستهلك energy. Look to this simple figure. You have two substances, A and B, bind together. This is anabolism, anabolic reaction to create big substance, large molecule, which is C. Other version, C will be broken down by catabolism and go into two substances, A and B. This will release energy. Anabolism will consume energy, تستهلك, require energy. How the energy is transferred, mediated by adenosine triphosphate, triphosphate, 3-phosphate, 1, 2, 3. Okay, so when we say triphosphate, it means that the adenosine molecule, this one, has 3 phosphate units. We call it ATP. Now look, this is the ATP, how it is formed. We have adenosine monophosphate, يعني phosphate وحدة. When you add to it another phosphate, it will become adenosine diphosphate, يعني 2 phosphate. If this substance you add another phosphate, it becomes adenosine triphosphate, يعني 3 phosphate. On the opposite, if you remove phosphate from the triphosphate, it will become adenosine diphosphate. If you remove another phosphate, it will return back to monophosphate, adenosine monophosphate. This is just simple diagram to see the different types of adenosine. Now the differences between catabolism and anabolism, both occur in cells. Catabolism reaction release energy. Anabolism require energy. تأخذ تستهلك. The catabolism reaction break bonds and energy will be released. Anabolism, a creation of bonds, and this will require, takes energy. Now, catabolism, larger molecule will be broken into small molecule, and this is called degradative reaction. التكسير. Large molecules are broken, while anabolism, smaller molecules are bonded together to create larger molecule. This is called biosynthetic reaction. تركيب المواد, biosynthetic reaction. Biochemical reactions, they have different materials. Starting material that start the reaction, and intermediate products, intermediary, we call them, and end product. These are just terms that we might use them during our course. So starting material, intermediate materials, and end products. The common metabolic processes are the following. The main one, aerobic respiration, and we will talk about aerobic respiration. Fermentation, this is anaerobic, without oxygen. Aerobic, it means with oxygen. Anaerobic, without oxygen. It is called fermentation, and there are other types of reactions. So we are going to talk about aerobic respiration and fermentation, which is anaerobic reaction. Look to the biochemical pathway. This is the starting material. E is the ending product, end product. And in between, B, C, D are intermediary. So starting material will be acted upon by enzyme number 1 to transfer it to B, acted upon by enzyme 2, which is specific to B, and so forth until you get the end product. So starting compound, end compound, and in between are the intermediaries, products or compounds. We come to the aerobic respiration of glucose. Aerobic respiration يعني هون احنا بنتكلم على عملية تمثيل استهلاك الجلوكوز لغاية شو؟ لإنتاج الطاقة. شو هي الطاقة؟ ATP. فلو ناخذ مولكيول جلوكوز واحدة، قديش الطاقة تنتج؟ قديش ينتج ATP؟ هذا هو اللي نتكلم عليه. فإذا الجلوكوز will be metabolized through 3 different processes. The first one is called glycolysis. تكسير الجلوكوز, glycolysis. Then a Krebs cycle. I am sure you have heard and learned the Krebs cycle. And then electron transport chain. That happens in a human being, also can happen in microbes. So glycolysis, Krebs cycle, electron transport chain. So aerobic reaction, look here, glycolysis, Krebs cycle, and electron transport chain. This will release about 36 ATP molecule from one glucose unit. Anaerobic respiration, no oxygen, start with the glycolysis also, but go into fermentation. Example, alcoholic fermentation, lactic acid fermentation. Now, the energy that is released from anaerobic respiration will be 2 ATP only from one glucose unit. Anaerobic respiration تنتج 2 ATP فقط، بينما الايروبيك ريسبيريشن 36 ATP، يعني 18 مرة أكثر. 18 folds more efficient aerobic respiration in releasing ATP than anaerobic respiration. Both start with the aerobic and anaerobic with the glycolysis. Glucose changed to pyruvic acid. This is called a glycolysis. 2 ATP only will be released from this breakdown of glucose to pyruvic acid. Pyruvic acid will be used by aerobic respiration and will release 36 ATP, as we summarize. We will go into details of this process. If pyruvic acid goes into anaerobic fermentation, no oxygen, it will release lactic acid. And only 2 ATP will be released from this reaction. While here we can get 36 ATP from availability of oxygen. So you can see here glucose plus oxygen, it will release CO2, water, and energy. Energy, what energy? In the form of ATP. Let us start with the glycolysis. As we said, both reactions start with a glycolysis, called the glycolytic pathway. Sometimes it is named as Embedden-Meyerhof pathway, the scientist who discovered it. Embedden-Meyerhof, sometimes called Embedden-Meyerhof-Parnas pathway. So you should know these terms. Is 9-step process, require specific enzymes. The end of glycolysis of glucose will be the release of 2 pyruvic acid, pyruvate. Each pyruvic acid has got 3 carbons. يعني ينتج بنهاية الجلايكوليسيس pyruvic acid، 2 pyruvic acid أو pyruvate. كل pyruvic acid ب3 كربون. سهلة جدا. Little energy only. 2 ATP will be released from glycolysis. وطبعا 2 ATP غير كافية لنمو المايكرو أورجانيزمز. نحتاج طاقة أكثر. This can occur, the glycolysis, in prokaryotes like bacteria and eukaryotes. If we are talking about eukaryote, like fungi are eukaryotes, human cells are eukaryote, animal cells are eukaryote. There is a reference here for you if you would like to go to net and have more information about this point. It's very interesting thing. Look here, glucose goes into glycolysis through enzymes. At the end, you will get pyruvic acid. This process has 9 steps. The end will be pyruvic acid, 2 pyruvic acid. Krebs cycle also called citric acid cycle. A tricarboxylic cycle, also another name. It's 8 steps. In glycolysis, we said 9 steps. Each step with a specific enzyme. How much energy we get from a Krebs cycle? Only 2 ATP molecules will generate 2 ATP. I am not teaching you Krebs cycle. I am teaching you the energy released from these processes that the microbes require. منين تجي الطاقة للميكروبس؟ أول إشي حكينا الجلايكوليسيس، ثاني إشي الكريبس سايكل. جلايكوليسيس يجيب لنا 2 ATP، الكريبس سايكل يجيب لنا 2 ATP. أما تفاصيل الكريبس سايكل هذه تأخذوها بالبايوكيمستري. At mitochondria in human cells, for example, but in bacteria at the inner surface of cell membrane, mesosomes. So this reaction, Krebs cycle, occurs at mesosomes of bacteria. Bacteria has no mitochondria, as you know. Bacteria has no mitochondria. شو بديل الميتوكوندريا and البكتيريا? Structure called mesosomes, which are present in the inner surface of the cell membrane of bacteria. You know, bacteria has got cell membrane وفي تعرجات هيك زي الحويصلات تسمى mesosomes. ممكن مراجعتها أخذتوها في المحاضرات الأولى. Krebs cycle starts with acetyl coenzyme A, combine with oxaloacetate to produce citric acid, a tricarboxylic acid. That is why it is called citric acid or tricarboxylic. You can see it here in the figure. Pyruvate now will be broken down. Pyruvate اللي حصلنا عليه من glycolysis will be acted upon by enzyme, and you get acetyl-CoA. Acetyl-CoA will bind to oxaloacetate, okay, and you get citric acid, right? And you can see here in this process, hydrogen will be released. 1, 2, 3, 4, 5. And here we will get hydrogen from this. So you get source of NAD edge later on. And one of them will be FAD edge. Not all of them NAD edge will be created. One of them will be FAD edge. We will come to this point in a moment. The last process in the aerobic respiration, electron transport chain. This is very efficient process, series of oxidation reduction of process. Energy is released by electron transfer. Electron transfer when you روح to end oxygen, end product, you remember, end oxygen. Final terminal electron acceptor, يعني يتقبل الالكترون terminal اللي هو الاكسجين.
دكتور الشاشة سكرت دكتور.
نعم.
الشاشة سكرت تبين عندنا خضراء.
نعم.
الشاشة تبين خضراء عندنا.
السلايد شو تبين؟
السلايد أخضر.
السلايد مغطى يا دكتور.
نعم.
السلايد تغطى بلون أخضر، مو مبين إشي منه.
هسه؟
هيك تمام.
هيك تمام.
طلع هيك هلا قبل شوي ما بعرف فد إشي نوت، يعني أنا كبست عليها راحت يعني. هسه تمام؟ Okay. Enzymes involve cytochrome oxidase. أحيانا نسميه cytochrome C or oxidase enzyme. transfer electron to oxygen. Oxygen هو الفاينل acceptor. Example, oxidase positive gram negative bacilli. Some of the gram negative bacilli are oxidase positive. This is how we are going to transfer it into practice. Okay, electron transport chain going to involve 10 NAD edge اللي جايتنا from glycolysis والكريبس cycle واثنين فات حكينا from Krebs cycle. تمام. الناد ايج 10 كل واحد تضربها في 30 اي تي بي ويجينا 4 اي تي بي from فاد 2 في 2 4 فاذا راح نحصل على 34 اي تي بي. عندنا 2 من glycolysis يصير عندنا 36. فإذا أنا هسه in conclusion as many as 38 molecules will be produced by oxidative phosphorylation. That is to say the transfer of ADP to ATP. ADP to ATP from glucose catabolism through glycolysis, Krebs cycle and electron transport chain. بينما الفرمنتيشن حكينا only 2 ATP ينتج. يعني الايروبيك ريسبيريشن 18 الى 19 مره اكثر، او الفرمنتيشن 18 الى 19 مره اقل من الايروبيك ريسبيريشن. Now in eukaryotic cells 36 to 38 ATP زي ما حسبناها. 36 إلى 38 are produced from catabolism of one glucose molecule. What is the meaning of oxidation? Oxidation, loss of electron. Phosphorylation, conversion of ADP to ATP. Phosphorylation, conversion of ADP to ATP. Adenosine diphosphate to adenosine triphosphate. This is called phosphorylation. Oxidation, loss of electron. راح نجي عليها بعد شوية. Now, chemical equation of aerobic respiration, you can see glucose with oxygen, ADP, phosphate ينتج لنا water, CO2 و38 ATP. تحول ال ADP إلى ATP. يعني خلاصة الكلام الجلايكوليسيس بروكاريوتيك يعني بكتيريا، ايوكاريوتيك انيمال سيلز فانجاي. البكتيريا من الجلايكوليسيس يجينا 2 اي تي بي، كريبس سايكل 2، الالكترون ترانسبورت حكينا 34، قديش صار التوتال؟ 38. بالايوكاريوتيك للمقارنة يعني مع الفنجاي او الانسان نفس الشيء، جلايكوليسيس 2 اي تي بي، كريبس سايكل 2 اي تي بي، الكترون ترانسبورت تشين 32 الى 34، المجموع 36 الى 38. يعني خلاصة الكلام انه الالكترون ترانسبورت تشين is the most efficient part of aerobic respiration، ينتج 34 اي تي بي، بينما الجلايكوليسيس والكريبس سايكل فقط 2. تمام؟ Fermentation. Now we go to the anaerobic metabolism of glucose. Oxygen is not involved here. Anaerobic. First step is glycolysis. حكينا أنه الجلايكوليسيس مشتركة بين الأيروبيك والأنيروبيك. You get from glycolysis pyruvic acid. Pyruvic acid, the end product will be only 2 ATP. حكينا بالفرمنتيشن فقط تنتج 2 ATP. وهذه الفرمنتيشن ممكن يستفاد منها في الصناعة. يعني مثلا عندك استخدام الخمائر، ييست، سكرومايسيس، أم بكتيريا، زيموموناس، هذه تستخدم في الصناعة. convert pyruvic acid to ethyl alcohol، تصنيع الإيثايل الكحول by these microbes. convert pyruvic acid to ethyl alcohol. ممكن البيروفيك أسيد هذا تستخدم اللاكتوباسيلاي ويتحول to lactic acid which is in manufacturing yogurt، صناعة الألبان، اللبن. بس ممكن تكون مضرة. ستريبتوكوكس تأخذ البيروفيك أسيد وتعملها لاكتيك أسيد. لاكتيك أسيد in the mouth will damage the enamel of the teeth. enamel يعني غطاء الأسنان. So the teeth will be damaged by microbes called streptococcus. streptococci الجمع. streptococcus المفرد، streptococci الجمع. We come back to the oxidation reduction. حكينا خلال كلامنا oxidation و reduction. Oxidation reduction called redox reaction. تعلموا من نقول redox reaction معناتها oxidation reduction. Oxidation, loss of an electron. حكينا oxidation, loss of an electron. Reduction, gain of an electron. انتوا تعرفون الالكترون charged negatively. شحنة الالكترون negative. So if a material gain electron تكسب electron الإلكترون هذا شحنته سلبية. The electrical charge of this material will be reduced. تقل الشحنة الكهربائية. That is why it's called reduction. So oxidation is loss of electron. Increase in the charge of the material because losing electron, which is negative. While reduction, gain electron. Electron is negatively charged. Therefore, the electrical charge of the material will be reduced. I think this is clear to you now. Dehydrogenation reaction, dehydrogenation, removal of hydrogen. A Krebs cycle you have seen in the figure how hydrogen is removed to make NAD edge or FAD edge. I have put to you video here for electron transport chain. It's very nice video. If anyone interested in this process, you can see video only for a few minutes. You can see here simplified figure for oxidation reduction reaction. Compound A will lose electron. This is electron negatively charged. This is called oxidation. Then the electron will be accepted by compound B, and because electron is negatively charged, the charge of compound B will be reduced. This is reduction process. Very simple oxidation reduction reaction, how illustrated. Let us go into anabolism. We have spoken about catabolism. تكسير الجلوكوز مثلا حكينا عليها. هسه anabolism require energy to create bonds. Energy provided by simultaneous catabolic reaction. Simultaneous يعني في آن واحد. يعني كمية الكاتابوليزم تساوي كمية الانابوليزم. Example عندنا 2 مونوسكرايد راح يرتبطوا مع بعض ببوند. شو يصير عندي؟ دايسكرايد. Similarly for a protein, for a nucleic acid. Now this organic photosynthesis, for example, photosynthetic material, and chemosynthesis is exactly also can do anabolism. Chemosynthesis either by light or by chemicals. Let us start with photosynthesis. Light energy will be converted to chemical energy in the form of bond. كيف الميكروبس تصنع. Photoautotrophs, example, photo, light, auto, it means CO2, cyanobacteria. كيف السيانوبكتيريا تصنع الطاقة؟ بهذه الطريقة. Photoheterotroph, some bacteria حكينا. Photo, light. بينما أغلب البكتيريا chemoheterotroph, chemical, مو light. The light will be trapped energy, تخزن الطاقة. This will be creating chemical bonds in the form of ATP. This will create bonds in the form of ATP and in the glucose. Bonds in ATP and in the glucose. Now the glucose will be broken down also, and more ATP will be released by aerobic respiration. So light will trap energy, and this energy will be transferred to chemical bonds in ATP and glucose. Glucose will be broken down and more ATP will be released. Some bacteria are oxygenic, photosynthetic bacteria, oxygenic, produce oxygen, photosynthetic bacteria. Anoxygenic bacteria, these are purple, green, sulfur bacteria. These are obligate anaerobic, photo light, auto CO2, photoautotrophs, and these bacteria use sulfur and hydrogen. Anoxygenic bacteria. يعني احنا لا تنسوا احنا بدنا نتكلم بانترودكشن الان، بدنا نتعلم تعابير معينه، يعني كلمات معينه. When we say oxygenic bacteria, they produce CO2. Anoxygenic, without oxygen, it means they are obligate anaerobe. photoautotroph مثلا ما تحتاج أكسجين ولا تنتج أكسجين، تتعامل مع السلفر ومع الهيدروجين. هسه سؤال question. Photosynthesis is the reverse of anaerobic respiration, of aerobic respiration. Photosynthesis is the reverse of aerobic respiration. Why؟ هسه نشوف. الفوتوسينثيسيس CO2 plus water light ATP ينتج شو عندنا؟ glucose, oxygen, water, ATP plus phosphate of course. فإذا CO2 water ينتج لي glucose and oxygen. Now if glucose through glycolysis plus oxygen, it will release at the end CO2 and water, one opposite the other. Okay? يعني if you have now glucose plus oxygen, this is aerobic respiration, it can break it down into CO2 and water. If no light, if the microbe live without light, they can survive anaerobically by fermentation. إذا ما عندك light وما عندك oxygen, الميكروأورجانيزم كيف تعيش؟ anaerobically by fermentation. If no CO2, they become photoheterotroph. Photo light, hetero, it means they use organic material as a source of carbon. Let us go into chemosynthesis. We have spoken about photosynthesis, now chemosynthesis. Chemo, autotroph. Chemo, chemicals. Auto, CO2, it means. So chemotrophs use CO2. Example, primitive bacteria. Archaea. I am sure you have heard in the first lectures about archaea. The archaea, some of them are methanogens. They produce methane, غاز الميثان. Archaea methanogens. How? Hydrogen plus CO2, they produce CH4. CH4 is the methane, غاز الميثان plus water. This is how the chemosynthesis of certain microbes, of certain organisms. Chemoheterotrophs, chemo, they use chemicals and the carbon source from organic material other than CO2. Most bacteria like that. Fungi, protozoa, animal cells, human cells are chemoheterotrophs. We stop here, and I hope that the lecture is clear to you, simple, and we are going to build on it the next lectures إن شاء الله. يوم الأحد إن شاء الله نبدأ بالبكتيريال genetics. Any question? Any question? Any student have any question?
الله يعطيك العافية دكتور.
الله يعافيكم عيني.
يعطيك العافية دكتور.
شكرا جزيلا عيني، الله يعافيكم. إن شاء الله تكونوا استفدتوا من هالمحاضرة هذه.
ممكن سؤال لو سمحت؟
إيه يعني تفضل.
هسه الستبس تاعت الجلايكوليسيس والكريبس سايكل بنحفظهم ولا ما فيش داعي؟
لا الستيبس هذه ما تحتاجها أنت. احنا بس نتكلم على كيف الطاقة تنتج، يعني بالعموم ده نتكلم عليها. أما تفاصيلها الستيبس هذه بايوكيمستري، إن شاء الله تكونوا ماخذيها أو راح تاخذوها بالبايوكيمستري.
تمام، شكرا دكتور.
شكرا. شو بعد سؤال أو استفسار؟ هل ستودنت؟
دكتور لو
نعم.
سمحت،
نعم.
الحين if no light ليش البكتيريا survive anaerobically؟
إذا ما في أكسجين، يعني تصور ظرف معين المايكرو أورجانيزم تعيش انيروبيكلي. أعطي لك مثل كلينيكال. واحد مكسورة إيده ومجروح ومكسور العظم، وتم وضع هذا الجبسونة ومع شد قوي، وبعد أكسجين ما يجي للمنطقة، هذه anaerobic condition. فcertain microorganism will use fermentation لأنه ما في أكسجين. تمام، واضح هذا؟ أنا حولت لك إياها إلى clinical example على اعتبار أنتم أطباء. واضح هذا المثال؟
واضح.
تمام.
دكتور بس
بعد
انه
سؤال.
المكتوب السلايدز if no light مش if no oxygen، انه ليش صار بدون لايت صار anaerobic؟
If no oxygen, the microorganism will go into fermentation.
دكتور فاهم فكرة انه بدون اكسجين anaerobic، بس لما ما يكون في
Anaerobic.
لايت اصلا، ليش لما يكونش ما في لايت ليش بده يصير anaerobic؟
لا، اللايت مسألة أخرى. يعني اللايت if
هي هيك
you
مكتوب
don't
بالسلايدات،
have-
السلايد اللي قبل
if you
انت
don't
فيه.
have light, the microorganism going to use chemicals instead of light. Right?
دكتور بس السلايد اللي قبل اللي انت فاتح عليه مكتوب اخر شيء if no light، اللي بعد هذا تحت. لا دكتور السلايد رقم 35.
نعم، هذا السلايد. If
آخر شيء
no
تحت.
light, yes, survive anaerobically by fermentation. يعني إذا أنت
شو
ما
العلاقة
عندك
بين الضوء والأكسجين؟
إذا if no light يعني ما في طاقة، منين تجيب الطاقة المايكرو أورجانيزم؟ فلا بد أنه تتحول to survive anaerobically. Anaerobical, this will provide the energy by fermentation ATP. على الرغم من قلة ال ATP اللي ينتج عن طريق الفرمنتيشن اللي هو 2 ATP only.
تمام وصلت. شكرا دكتور.
نعم. If no CO2, look to the, they become photoheterotroph. نعم عيني.
شكرا دكتور.
على عيني، يا أهلا وسهلا. إن شاء الله تكون واضحة، سمبل، تقروها قراءة سهلة إن شاء الله. وأي سؤال عندكم أنا حاضر. من راح يكون عندنا المحاضرة المرنة إن شاء الله الأسبوع القادم بعون الله. ناخذ محاضرة أخرى، ونهاية الأسبوع إن شاء الله راح يكون عندنا محاضرة اسمها محاضرة مرنة. أي أسئلة عندكم بعد ما تقروها وتفهموا المحاضرات تبع اليوم وتبع الأسبوع القادم، إن شاء الله نجاوب على أسئلتكم كمان. أوكي؟
يعطيك العافية دكتور.
All the best عيني. يا أهلا وسهلا. فإذا we conclude this lecture and we end it. See you again, inshallah, next week.